Okay, so today we're going to be talking about topic 6.3, which is all about the immune system. So, as you know, there are probably millions of microbes and pathogens floating around the air right now that could be colonizing your body for materials and growth nutrients. And so, the language that we're going to use today is that we're going to call these guys collectively pathogens, right? So, pathogen is any organism that can cause a disease of any kind, and they include things like bacteria, such as E. coli or salmonella, that you might encounter during food poisoning, or viruses such as herpes or the flu or the cold viruses, or even protists, little critters like the protist that causes malaria, or fungi. So all of these entities, some of which are living and some which are not, are able to cause disease, and we collectively refer to them as pathogens. So the reason why you are probably not dying from one of these horrible diseases is because of the action of your immune system. And so there are multiple layers of how our bodies protect ourselves against the invasion by these pathogens, and they come in two flavors. So one is the innate, which is something that is always present. It's the immediate, non-specific response in our bodies. And so this is where we'll start talking our discussion today. But there's also the adaptive, or also known as the acquired immune system. So compared to innate, this one is a delayed one. And rather than being non-specific, the adaptive acquired immunity is very specific to a pathogen. And in contrast to the innate immunity, which is an immediate response, the adaptive or acquired immune system is more long-term. It's geared towards repeated exposures. So let's go ahead and start with the innate immune response, and we'll talk about our first line of defense, right? So the first line of defense is really all about blocking entry of a bug, of a pathogen into the body itself, right? And so this involves things like the skin, which acts as a barrier between our internal organs and the rest of the world. And it can also include things like the mucus that is in our throats, which act to trap pathogens that do somehow make it into our, say, air passageways. And it can include things like secretions that we have in the eyes, which will actually digest these pathogens if they actually do land on them. So things like tears or even your saliva are going to be present in are going to have enzymes present in them that are specifically there to digest a large swath of different kinds of pathogens. So this first line of defense is part of the innate immunity. So it's something that's basically always present, never absent, and again, it's non-specific, so it's a very generalized response. So what we've mentioned so far is the external innate immune system. But we do know that injuries do occur, right? And so we also know that for most of us, if we get a cut, we don't necessarily bleed to death. Our body actually has responses to that as well. And so what keeps you from bleeding to death, you probably already know, is a blood clot, right? And so if you happen to get a scratch from your cat, or you haphazardly scratch yourself on like a door hinge, you're not going to bleed to death because something is happening that is blocking that wound. And you probably also notice that when these events happen, you get a scratch of some kind, that the area becomes really red and swollen. And what's happening there is that not only was the site blocked somehow, but there's an immune response happening in the immediate aftermath of that injury. And you don't have that scratch forever, because what is also happening in response to an injury is that that area is stimulated to produce new cells to repair that tissue that was damaged, right? So blood clotting, 3 responses. Number 1, keep you from dying, block the site of injury via blood clotting, then recruit immune cells to fight any pathogens that might have gotten in, and then 3, once you have those 2 things taken care of, you stimulate new cell growth to repair that tissue. So let's take a look in depth at what is occurring during blood clotting. So we just talked about the circulatory system, and we know that there are vessels all over your body. They include things like arteries and veins and capillaries, and that within those vessels are for sure red blood cells, which, just to call back to our earlier topics, the red blood cell's primary job is to carry around oxygen via that hemoglobin, which is also that protein that is mutated in sickle cell anemia, right? But anyway, you have vessels with your red blood cells. So this is stuff that we have talked about. What we are going to add here is a new player which is also around in these vessels, which is something called platelets. So platelets are basically fragments of old red blood cells. So they are not red blood cells themselves. They are a totally different class. So these platelets are the main player in what is happening during blood clotting. So the platelets, once an injury is occurring, what they will actually do is they are the first to rush to actually block the wound and to stop that bleeding, right? And the second thing that a platelet will do, again, this is not red blood cells, this is the platelet that is going to rush to the area to block that wound, and it is the platelets that release the first of many signals in a cascade that's going to result in the formation of a clot. So once the platelets rush to the area and they block the wound, they're going to release a clotting factor. And so we did talk about clotting factors in our genetic diseases. Remember, hemophilia is a deficiency in one of these clotting factors. So that would lead to uncontrollable blood loss in the person. So that's where this is coming back into play, right? So platelets first block the wound, then they release some kind of clotting factor. And so what that clotting factor is actually going to do, and we're just kind of zooming in here to give ourselves more real estate, that clotting factor is going to be responsible for converting a protein called prothrombin, which is inactive, right? And so basically it's going to convert that prothrombin into thrombin, which is now active. So really this clotting factor you can think of as like an enzyme. It's coming over here and it's cleaving this prothrombin to thrombin, and it is the thrombin that is active. And so once thrombin is active, it can now work on its main target, which is fibrinogen. So in its inactive form, fibrinogen is soluble. Once thrombin is able to act on it, that thrombin converts that soluble fibrinogen into insoluble fibrin. And it's that insoluble fibrin which is actually forming the mesh on top of the wound and is officially creating the clot, right? So from start to finish, you have the platelets, which first come here. Let's go all the way back. Once a wound is detected, they rush to that site, they block the wound, the platelets release a clotting factor. That clotting factor is then responsible for the conversion of prothrombin to thrombin, which is active. And then that thrombin can now convert soluble fibrinogen into insoluble fibrin, which is what creates the mesh in the end, right? And so once that mesh is there, then the clot is officially formed. So this is the full process of blood clotting that you do have to know. So another way of just looking at this same thing, we have our wound being created, though again it's the platelets. So in this graphic they're represented as like the little yellow things. The platelets are what is releasing the clotting factors. They're converting prothrombin to thrombin. That active thrombin is now converting soluble fibrinogen to fibrin, and it's that fibrin with all those red blood cells and the platelets that actually create the clot. So that's the entire process of blood clotting. So this is our first line of defense, again, which is part of the innate, right? Immediate, non-specific response to a pathogen. And so now let's talk about the second line. So if something makes it past either the skin or those mucous membranes or those secretions, or something gets in through a scratch that the clot is not able to address, then a pathogen may encounter a second line of defense. And so these are going to be referred to as leukocytes, also known as white blood cells, right? So we have our red blood cells, which are carrying oxygen. So the leukocytes are the white blood cells. And leukocytes sounds a lot like leukemia because that is, leukemia is the uncontrolled division of these white blood cells. So the main type of leukocyte that we're going to talk about is something called a phagocyte or a phagocyte. And so what these do is they endocytose pathogens. So they actually will, pinch off their membrane, surround a pathogen, chew it up to create an antigen, which is going to be important as a kind of trainer for the adaptive or the acquired immune system. So let's take a look at this second line response, these macrophages or phagocytes. So these phagocytic cells, what they're doing, and so this is an actual white blood cell. So this is an actual macrophage, and you can see that it's reaching out and kind of gobbling up these pathogens, right? So it actually reached out an arm, it's grabbing hold of them, it's dragging them inside itself, and you see these little vesicles here. So what these little vesicles are lysosomes, right? So if you think back to like our animal cell, the lysosome is fusing with the vesicle that contains the bacteria. So this little cartoon is showing it right here. So lysosome comes and the lysosome chews things up, right? It's digestive. So what's happening here is that this phagocytic cell is endocytosing, right, this pathogen. That vesicle with the pathogen is then going to fuse with the lysosome and it is going to be digested. So we call that process phagocytosis, right? So instead of endocytosis, it's phagocytosis because it's a phagocyte. It's inhaling that pathogen and it's basically chewing it up. So this is just a cartoon of that where you have the microbe entering. Again, this is being endocytosed here, right? So this would be one of our active transports. The endocytosis is where the membrane just pinches in. So we create a vesicle when we do that. So this is a phagosome. It will fuse with the lysosome to become a phagolysosome. And then the lysosome does its job. It's going to digest that microbe, and we'll talk about what it does with the parts of the pathogen that it created to make antigens next, right? So this is the main event of what is happening during phagocytosis. So again, pathogen meets a macrophage or phagocyte, is digested, right? And so what is created basically with the parts of that pathogen, right? Pathogens are made of proteins, they're made of nucleic acids, so it chews it up and then it presents that fragment of protein or that fragment of nucleic acids on its surface, and we give it a special name. When a pathogen is chewed up into a specific part that is presented, we call that an antigen, right? So antigens is any part of a pathogen, could be protein, could be nucleic acids, that was then presented on the outside of that macrophage, on the outside of that phagocyte. So the reason that it's doing that is it's telling the next part of the immune system, hey, this is what our enemy looks like. Here's a part of it. So you might also hear these phagocytes, these macrophages, as being referred to as antigen presenting cells or APCs. So that's what that's talking about. It's because they are the ones that are responsible for first engulfing that pathogen via the endocytosis, chewing it up during the phagocytosis, and then presenting that antigen, right? That little fragment that's going to inform the next line of defense about how can we defeat this invader, right? So that second line of defense is going to inform the adaptive immunity side or the acquired immunity, which is the third line of defense. So that third line of defense is all about the synthesis of antibodies, which are going to be specific to a certain antigen. And these antibodies are made by a different cell class called a lymphocyte. So the phagocytes were called leukocytes. Their origin is the white blood cells. So the lymphocytes, when consumed, originates from the lymph nodes, right? And so again, their whole deal is to make antibodies. So antibodies, also known as immunoglobulin, right? So those were one of the proteins that we learned about in like section 2.4. Their whole job is to either block a pathogen's attachment, interfere with its function somehow, because remember, this antibody is going to be specific to an antigen, to some part of that pathogen. So it'll either block it entirely, or it will make it more visible to our immune system to say like, hey, come take these pathogens out, right? So this would be the third line of defense. So where the innate, right, was always present, it's not specific. Now with the adaptive immunity, it is very specific to a pathogen. We're using the antigens that were created by those phagocytes in the second line of defense in order to mount a specific response to a pathogen, one that can also be long-term if there is a repeat exposure, for example, right? And so this is going to involve, again, the lymphocytes, whose whole job is to create antibodies. And some of the cell types that we're going to see are things like helper T cells, B cells, plasma cells, and memory B cells. So all of these are lymphocytes, and all of them are involved in the production of antibodies. So what happens after a phagocyte endocytoses and chews up a pathogen and creates an antigen? So let's wait for the animation to circle back around. So this is our phagocyte. It makes an antigen, and it's presenting that antigen. So who it is presenting the antigen to is a helper T cell. So this is one of our lymphocytes. When a phagocyte has an antigen to present, the helper T cell comes over and is like, okay, this is your antigen. What I'm going to do is then take your antigen and see, can I find a match for it in our arsenal of our adaptive immune system. So phagocyte digests our pathogen, creates these antigens, going to present that antigen on its surface, presents it to a helper T cell. That helper T cell will then go try to play a matching game with B cells. And so what I should point out is that there are millions of kinds of B cells, and each type of B cell has only one type of immunoglobulin on its surface, only one type of antibody that that particular B cell will make. When a helper T cell has that antigen, it's coming along and it's trying to find a match with the particular type of B cell that can bind the antigen that was created over here. It's not interested if this B cell can't make an antibody that's going to bind the antigen. So once it finds a match, and it can go through, thousands, hundreds of thousands, millions, right, of these B cells until it finds a match, a B cell with a matching antibody. And once it finds a B cell that does have those complementary antibodies, it will then activate that B cell to kick off the rest of the adaptive immune system. So once the match is found, right, and again, they can come in really millions of different combinations here, and they're all unique. So once a match is found, that B cell becomes activated because it's like, hey, I've got an antigen that you can help fight. So that B cell will convert into something called a plasma cell, which is going to secrete and make massive amounts of that particular antibody that are made to fight the antigen that this helper T cell found. So it will just produce massive amounts of this antibody that's going to go out and attach to that specific antigen that it brought in on the microbe. So there are two paths, right? Or just to reiterate the path that we saw, right? So helper T cell will look to find a matching B cell. If it finds a match, that B cell converts into a plasmid cell, which just releases a ton of antibodies because there's something in the body that needs to be flagged and tagged and stopped. So that B cell becomes a plasmid cell, releases tons of antibody, right? And what that antibody is going to do is it's going to run out and stick on the microbes, right? And so again, these are just the immunoglobulins, right? The antibody, which was made by the lymphocytes, is now going to go out and stick to the pathogen, which makes it very visible to these white blood cells, to the phagocytes. And it's like, hey, come eat this, right? So not only does it interfere with its attachment and function, it makes it much more visible to the rest of our immune system so that it can come and be attacked. So just to review the process from start to finish, that we have a pathogen that is initially engulfed by some sort of phagocyte. That pathogen is digested into little bits by phagocytosis, and then that antigen that is created from those digested bits is presented to one of the lymphocytes, which is the helper T cell. So that helper T cell will then become activated. It's going to go through its entire library of millions of B cells, each with a different antibody, until it finds a match. Once a B cell is matched, that B cell will convert into a plasma cell. It'll make a whole bunch of them because this is running around our body. We need to stop it, which is just going to turn your body into a factory of antibody molecules that are going to go out and stop the infection that are going to help your body recognize it and clear it out. What it's also going to do is create a memory B cell. Because if you see it once, chances are good you're going to see it again. And so this is the long-term portion of it. This is why vaccines are able to work, because those memory B cells will float around your blood for months, sometimes years, sometimes your lifetime, to be ready to turn right back in to plasma cells that are going to secrete massive amounts of antibodies that are going to fight that infection. So the two halves, phagocyte, digests the pathogen. This is what creates the antigen. Everything from that point forward is a lymphocyte whose whole job is to produce those antibodies which bind specifically to that antigen. Okay, so let's talk about an infamous disease of the immune system, HIV, which is human immunodeficiency virus, which is the virus that causes AIDS, which was called acquired immune deficiency syndrome. So the AIDS virus, or I'm sorry, HIV, which causes AIDS, which we didn't really even know existed in the Western countries until around the 1980s, was a very deadly disease for decades, and it still is in many parts of the world. So here we have a distribution of HIV, so this is still considered a pandemic. It is spread worldwide. So America is one of the more heavily affected areas, probably because we have a lot of drugs to treat this, right? And so you can see that certain areas of southeastern Africa are particularly infected. So HIV is a virus that infects those helper T cells, which are involved in the activation of that antibody production. So HIV is considered to be a sexually transmitted disease, but it can be transmitted really through any trading of bodily fluid, or like significant amounts, right? So things like sharing needles or blood transfusions. There's also transmission from mothers to fetus, right? And so what is happening is that HIV, remember viruses have these attachment proteins, is specifically attaching to the CD4 receptor that is on those helper T lymphocytes. And so if we remember, right, that the helper T cell is kind of this intermediary between the phagocytes, which is that second line of defense that's creating the antigen, and it is the in-between step of what is talking to the rest of the acquired immune system, the rest of that adaptive immune system. And that if we lose it, right, we lose all of that ability to produce specific antibodies to that pathogen. So if we have a situation where HIV is invading and killing helper T cells, then we not only lose the helper T cells, but every specific every adaptive immune response that we would have after that, any ability to really produce antibodies for anything, becomes compromised. And so HIV is going to result in reduced numbers of active lymphocytes, and the end result is that the affected individual is immunocompromised, and they will die from really any opportunistic infection that would normally be fought off in somebody who was healthy. So again, what we see is this HIV virus that is binding to the CD4 positive receptors on those helper T cells. And so this virus goes into a lysogenic phase initially. So if you think back, lysogenic means that I integrate into the genome. And so what is occurring is that the viral genome gets in there, integrates into the genome, and then will be latent for sometimes up to 10 years. And so the other thing that, and this is just frosting for you, that would occur after the integration in the genome is that a lot of patients were first presenting with Kaposi sarcoma, which was a kind of cancer. And that's because the virus has a really strong promoter, and sometimes it would land, in front of a cell, in front of a gene that was promoting cell division. And so that promoter was now driving these cells to divide, because it's really trying to drive, production of this virus. So once it is lysogenic, once it is integrated into the genome, it kind of hangs out for a long time. before becoming active at some later time. And so when it becomes active and starts to replicate, that is what is leading to the death of these helper T cells. And so when you look at the acute infection, right, you have this first dip in CD4 positive helper T cells that does start to recover in its latent period. And so the person may have like a little bit of a problem, but they kind of bounce back within the first, couple of months. And it's not until years later that they start to present with these really strange infections that they're starting to die from, and it is because the virus is becoming active and killing off their helper T cells, which is really impairing their ability to fight off infections. So that can affect really any tissue. So things that normally you would be able to fight off in a healthy immune system, that is why HIV is so deadly. So that is a wrap for 6.3.